Thank you very much, and it's my honor to be able to join you here today and to be back on the prairies. I was in North Dakota for 11 years, and I have a family that farms in Minnesota, so very familiar with this area of the country. And as we're talking about soil biology and soils helping to build resilience in the system, this is a very unusual November day here in Saskatchewan. So these are the types of resiliences that we're going to be trying to look at building in this system. And as was noted, I'm going to talk about many of the trials that we have going on at the Rodale Institute and many of the things that we're trying to do in order to be able to get information about how to be able to build resiliency in organic systems and what that resiliency is going to mean. So I wanted to kind of start off with sort of laying a foundation of what I'm meaning about building resilience. So we're going to build resilience against climatic uncertainty. I tell people that the only certainty that there is in agriculture is the fact that the weather is going to be uncertain. Everything else is pretty much an uncertain thing when you're in agriculture. And so we're really facing a lot of precipitation fluctuations, a lot of temperature fluctuations. Again, today is an obvious notice of temperature fluctuations. But we also need to build resilience for nutrient management, looking at the availability of nutrients. What's the difference between having available and unavailable nutrients, and how the biology can help with keeping that availability of those nutrients throughout the growing season, and the benefits of that as opposed to in a conventional system or in a system in which you're adding any types of synthetic or chemical types of nutrients, how that's going to actually cause very much fluctuations in the availability of nutrients. Because they're bringing on a lot of available nutrients, but not maintaining that availability throughout the growing season. So there's a lot with timing and availability there. I want to address some of the resilience that we're trying to build in helping to manage various types of pests. So looking at managing weed pests, insects, and diseases. And all of these things are doable with the way that we're going to be managing the system. And so that's what I want you to be thinking about, is basically this idea of regenerating soil health. I talked yesterday some about soil health, and the definition of soil health is the continued capacity to function. And so that's what we're really looking at, is these functions. And so when we're looking at this building of resilience, that is the functions that we're looking for soil to do, is to build the resilience against all of these different components that you have going on. So I wanted to sort of start off and kind of give you an idea of, some of these other fluctuations that we're seeing. This is a farmer that I've worked with in North Dakota when I was there. Oh, sorry, I'm looking at the wrong thing. So we're going to start off with climatic uncertainty. So here we are. This is a picture of a farmer that I worked with in North Dakota for a number of years. He is not an organic farmer. I want to lay that out right up front. He is a no-till farmer. Some of you may have heard of him, Gabe Brown. He is not an organic farmer, but he's utilizing many organic practices and cutting back tremendously on his inputs. So you're finding a lot of farmers out there that are on the path of becoming organic farmers and what that means. And he's looking at converting some of his acreage to organic agriculture and what's going to be involved in that. But this is sort of a good example of some of the resilience that we can build into the system. So even though he's not an organic farmer, he's utilizing organic principles of utilizing cover crops or green manure crops. He's utilizing animals in his system. He's building up soil health in that system. And so what he has found is, we had a particular rainfall event in 2009 where we had 13 inches of rain in a 24-hour period. And essentially this was the next day. We were out in his field. Nobody got mud on their boots when we were out there. You can see there's a little bit of a slope to his field. There's no pooling of water there, very little residue movement. And the reason that is that he's improved his infiltration rates over 20 years with doing these organic practices that has improved his infiltration rate from a ½ inch per hour up to 8 inches per hour. And these are things that we can do. When we're looking at building resiliency, this is what we're trying to do, is create these systems. And so as you build resiliency in this system, you're going to be able to manage water. much better. So what I wanted to really illustrate here is the soil that's in my hands. These are from plots that are side by side. This is from the plot that was the sunflower as the cash crop and had a mixture of plants that was growing in between as a companion crop that was in there. And essentially, you could see these are side by side, taken from the same area, and one is dry soil and one is wet soil. This is what the soil can do for us if we build the health of the soil in the system and get that inherent resiliency so that the water, when it falls, is going to go into the soil, and the water, when it falls, is going to be held in the soil for a longer period of time. And we've seen similar results. So this was in North Dakota. These are results from our farming systems trial, which is discussed. This is a trial that was started in 1981. So this is the 37th year of continuous production in that trial. 81 was a production year, and 2016 is a production year, so that's how we get to 37 years of production. So we've had 37 years of production in this system, and essentially it's a research trial in which we are comparing two different types of organic management systems to a conventional management system. And initially, we didn't have GMO varieties, nor did we have reduced tillage or organic no-till, rotational tillage, or rotational no-till practices. And we introduced those in 2008. So we are continuing to modernize this experiment and making sure that we are following through in the various types of practices that are very relevant to what's happening. So the organic manure-based system is about a 8 to 10 year rotation. It has a perennial phase in the middle of it, which is why it's an 8 to 10 year rotation. We've got a perennial phase that's either a 2-year perennial phase or a 4-year perennial phase that we're looking at. It has a diversity of crops. Most of those crops would be crops that are going to be used for livestock feed. So we've got both corn grain and corn silage. We have wheat and oats. And soybeans, all as part of that rotation in there. And then we've also got our organic legume system. Our organic legume system it's a 4-year rotation. It has corn, soybeans, oats, and wheat in that system. And basically what we're looking at with that is utilizing, because it's a shorter rotation system, and we utilize cover crops in there, our only fertility input into this system, we don't use any organic fertilizers in the system, we don't use any organic pesticides in either of these systems, so our only inputs actually come from what's in the system itself. So in the organic legume system, it is from the legume crops that we have in the rotation and the cover crops that we're using between our cash crops. We do grow a cash crop every year in this system as well. And in the organic manure-based system, we have an additional amendment of nutrients that comes from composted manure. And the amount of composted manure that we apply, this is a closed loop system, meaning that the amount of composted manure that we're going to be applying is calculated every year based on the previous crop's yield. So depending on last year how many head of cattle we could raise on the amount of grain we produced, that's how much manure that we could compost. So it's a closed loop type of system. We're not looking at getting any off-farm types of inputs to this. I'm going to kind of go through some things very quickly, but I do want to encourage you, if you want more information about these systems, more details into the backgrounds of this, we do have a number of things on our website, so I want to encourage you to go to our website. We have a 30-year report and some updated materials as well, being able to show what's going on, but I just wanted to go through and show the highlights and kind of give you some information so you have it in context when I'm talking about these different systems that we have. And so what we saw with these systems, again, we're now looking at climatic uncertainty as the resiliency that we're trying to build. What we found was that these are side-by-side plots. So the way that this is designed is we just have a 5-foot buffer between, it's on a 12-acre field, we have a 5-foot buffer between the individual management systems. This was designed in 1981, so it was before we had organic standards where you need to have a 35-foot buffer between the systems. Right now, we just have a 5-foot buffer between the systems. But when we're taking our samples and doing our analyses, we take it from the middle of our plots, and our plots are 20 feet wide. So essentially, we're getting that 30 to 35-foot buffer between each of the systems by where we're taking our samples. But what's interesting to note, again, these are pictures taken from the same day. This is wheat fields after a rain event, and you can see the water that's pooling on the surface in our conventional system and less water that's pooling on the surface in the organic system. So we're getting better infiltration rates. We're improving the ability for the soil to absorb the moisture, to absorb the rainfall when it occurs. And the reason that that's happening is that we're increasing the porosity in the system. Biology is driving this. Biology is important to creating, they engineer the soil environment. They create what are called soil aggregates. They're little pellets that are in the soil, and by engineering that environment, it creates, they bind soil particles together in little balls, and then in between those little balls is open space, and that's the porosity that's there. And so as you improve porosity in the system, you can improve the infiltration rate for the first inch by 167%, and for the second inch, which is even more important when we're facing these climatic uncertainty where we have heavy rainfall events, we no longer have day-long soaking rains. Those are going to become even more and more a rarity. It's going to be heavy downpours and prolonged periods of drought between that. That's where we're heading in the near future. So we increased the infiltration rate of the second inch by 650%. So we're actually being able to hold more water that's in the system. The other thing that happens in addition to increasing porosity is the fact that we're also increasing organic matter in those soils. And as you increase organic matter in the soil, you increase water holding capacity. The structure on a chemical level, the structure of organic matter allows for it to be able to have open space in the molecular structure that's going to allow for the molecule to act a little bit more like a sponge, so that you have this open space on a macroscopic scale where you're having organic matter that's going to be able to hold on to water for a longer period of time and be able to do that level of absorption. The other thing that we're finding, and this was actually some research that was done in the 1950s, hasn't really been discussed too much. It's been discussed a little bit more among organic agriculture and sustainable farming practices. But basically what they found was that unfertilized corn needed nearly 5 times the amount of water as fertilized corn in order to produce the same yield. And so what they looked at, and the chemical companies have still used this as the way of basically selling fertilizer, that you need to add fertilizer if you want to be able to enhance your yield. Now again, this was done in the 1950s, so genetics and various types of things have changed some of this, but the reality is one of the things that we need to be thinking about, and this is part of looking at this building resiliency in your system, is changing some of the paradigms. Now, I know all of you as organic farmers have changed a lot of paradigms in your world already, and in comparison to many of your neighbors. But part of the things that we need to be looking at in changing our paradigms is where we're thinking of fertilizer coming from. So in a conventional standpoint, the fertilizer is coming from conventional chemicals. In an organic standpoint, is the fertilizer, is that fertility still coming from something that's off the farm? How much fertility is actually coming from within the system itself? How well are we utilizing that natural fertility in the system and utilizing the biology in the system in order to be able to enhance what's going to be happening? So looking at being able to shift some of those paradigms that we have. So now we also saw issues when we had too little moisture. Around the early 1990s in Pennsylvania, we experienced a drought. And what we found, again, these are our side-by-side plots. So we've got our organic plot here, our conventional plot here. We've got our 5-foot buffer in here. And what we found in the organic plot, you could see that this was doing much better. This conventional corn is actually planted 3 weeks earlier than our organic corn. And it's because of the practices we use in planting the organic corn, that we're going to be planting it a little bit later. So typically we plant a longer day, we're going to harvest at the same time, typically. I say typically because this year our organic is a little bit behind our conventional, and I'll explain why in just a minute, but typically we're harvesting them at the same time, so we actually use a longer day variety of conventional corn and a shorter day variety of organic corn. Typically that also means that our organic corn should automatically yield less than our conventional corn does. Typically, automatically, right? These are paradigms. These are the things that people think about when they're making these selections. These are the choices that are made based on crop scientists and crop breeders in saying that this is the way it should act. But if we utilize a biologically based system, we can go beyond the performance of what people normally would think should happen in that system. So you can obviously see that our yield was higher. It was about 30% higher in the organic system than in the conventional system, and we had more water infiltration in the organic system than in the conventional system. Now, when the system first started off, our farm manager was actually very, very concerned because initially the conventional corn it was almost like this picture was reversed. The plants were a little bit smaller, but it was almost like the picture was reversed. The conventional corn looked really, really good, and the farm manager was a little bit tempted to go out there and switch the signs on which plot was which. But again, we're building resiliency for the long term. What happens in the spring when the crop first germinates does not necessarily entirely dictate what's going to be happening throughout the growing season and what's going to happen in the fall, when it is that you're going to be going to harvest. And so, again, many farmers are going to go out there and you're going to be making decisions based on something you might see very early on in the growing season, whereas the system, because it has built within it innate resiliency, it's still going to be able to thrive. It may do it a little bit slower, it may start off a little bit more slowly, and it may start off not looking quite as well as you would like to have it look, but in the end, it's actually going to start to perform much better. Now, this is what we saw in 2015. And in 2015, again, these are side-by-side plots, our organic corn and our conventional corn. In these two plots, the organic corn here yielded about 18% higher than the conventional corn. We were under a county-wide drought at this time. Our conventional corn started to dry up in late August, just like everybody else as you were driving around. This picture was taken September 13th, and you can see that we didn't have a drought in the organic plots. Now, as a research scientist, I got to tell you a little bit secret. We actually put up these little invisible barriers that you don't see so that the clouds will fall on one side and not on the other side. It's amazing what 5-foot buffers can do. I've seen the same thing happen in many roads as I'm driving around in the prairies. You'll go and drive around fields and it's like, one you're on planet Earth, one you're on planet Mars. I don't know how that happened, but somehow a road changed the way that precipitation fell. That doesn't happen that way. This is what it is that we're seeing in this system, that we're building up the resilience. There was no reason to have to file anything with the government as far as saying that we were having or get crop insurance to cover our organic plots, because we had that inherent resilience in this system. Now this is 2016. Picture's a little bit dark here. I apologize. I should lighten it up a little bit more. This was 2016. Again, organic plot, conventional plot. Similar type of thing that you're seeing. This response is continuing to occur. We've got much taller corn in the organic corn, much less drought stress. So we're starting to see where we're going to be able to get more moisture into our soil and hold on to the water for a longer period of time and build up that resiliency that we're looking for in the system. And again, what's going to be driving this is biology. It's the formation of these soil aggregates. And you can get biology to respond in a very, very short time window. So the pictures that I have here are all aggregates that were formed in less than 3 months. In fact, these aggregates that are here were actually formed in just a little over 30 days. So you can get a very rapid response in biology if you create the appropriate environment. And we talked about yesterday the fact that the organisms in the soil are basically starving and homeless. Your job is to feed them and to make sure that they can maintain their habitat. This is their habitat. This is their home. Guess what happens when you till up the soil? Guess what happens if you don't keep the soil covered by some sort of living plant or keep the soil covered by some sort of mulch or crop residue? The impact of raindrops hitting the surface of the soil in just a gentle rainstorm, the amount of energy has been measured with that and is equivalent to 20 tons of TNT. Little explosions are happening on the surface of the soil that's breaking up these aggregates every time it rains. If it is that we don't protect the soil. That's all that these little creatures want us to do. In a handful of soil, in a handful of healthy soil, there are more organisms than the number of people who ever lived on planet Earth. Well over 10 billion organisms, and all they need from you is food and a nice place to live. And you don't even have to build the place for them. They do it themselves. All that you have to do is make sure you don't destroy it. So these are the things that we're trying to do. And the thing here, this one that's glowing green, this is actually after staining it in the laboratory. What we're looking for there is a biomolecule produced by Arbuscular mycorrhizal fungi, by organisms in the soil that connect the plant roots to minerals that are in the soil and help to transport those minerals inside the plant roots. They actually help to engineer these aggregates. They form the framework for the aggregate structure. They basically are that building frame that you have there. And then they make a biomolecule, a protein called glomalin, and that glomalin creates sort of like a waxy seal on the surface of the aggregate. So that when the aggregate gets wet, like we're going to do here, when the aggregate gets wet, if it has glomalin on it, it will stay together. If it doesn't have glomalin on it, it'll fall apart. So what we have here is we have soils. This was from research that I did in North Dakota. Again, unfortunately not on an organic system, but the similar types of results we've seen with our organic soils as well. But we've got these aggregates, same soil type. Plots are all separated at most. This moderately grazed pasture was about a half-mile away from where we had the farming research plots, where we had a conventional till spring wheat fallow system, a no-till spring wheat, winter wheat sunflower system, and a moderately grazed pasture. So at the top, these are soil aggregates. All I did was I passed the soil. You can do this at home if you want. Get 2 different types of screen of 2 different sizes. So, you know, some window screen, some different types of insect screening that you have of 2 different sizes. The soil that passes through one screen, collect that, put it through the next screen. Whatever stays between those 2 screens, in this case, I had a 1 millimeter screen with 1 millimeter openings and a screen with 2 millimeter openings. So I have 1 to 2 millimeter size aggregates. Whatever stayed between those screens is what I collected. And then I just put it into a glass dish and I just added water. Now I sprayed the water. I used a little spray bottle and I sprayed the water, and I only let the water hit the edge. You don't want the water to spray over the aggregates themselves, because that's like your little TNT bombs that you're going to be dropping on them. So I had the water just slowly move into the aggregates, and then you can see the difference in the amount of aggregates that stay together. This number at the bottom, WSA, is just a percentage. It's called water stable aggregation percentage. So it's just the percentage of those aggregates that stay together. And so what we're finding is that, first off, when we reduce the amount of tillage that we have in the system, we're getting better aggregate stability. But if we include something where we have a moderately grazed pasture, or we have plants growing for a longer period of time, now we're going to be able to have a much higher rate of aggregate stability. And the reason that is because this little molecule that's on us outside, that's forming that weather sealant that's on there, that's making that protectant, that's something that the organism is going to make only if it has enough carbon resources to do that. So the first thing that the organism is going to do, that this fungus is going to do, its first job is to go out there and get minerals and water for the plant. So it's going to use the first amount of carbon that it has to extend its body to go out there. These fine threads that are its body, to extend that further out into the soil. The second thing that it'll do if there's enough carbon resources is it'll make that biomolecule. Now, in an annual cropping system where we aren't using cover crops, or if you're not using green manure phases in there, what you're going to end up having is basically not a whole lot of aggregate stability because there's not enough carbon inputs in a crop year to allow for the production of this glomalin. Again, you want to feed them and you want to help them to maintain their habitat. When we're looking, oftentimes too, you're looking at saying, you know, well, I can only grow short season crops. It's difficult for me to grow a cover crop. You don't understand. It's cold here, despite today. It's cold here. I do understand. I get that. But that can't be an excuse not to feed the soil. The soil is your most important resource. You need to start treating it as if that's what it is. Because if you don't feed the soil is not going to feed you. The plant doesn't care where it gets those mineral nutrients from as long as it gets those mineral nutrients. For you, the only thing that matters is who pays for it. You can either pay for it in keeping the soil fed and the organisms functioning it, or you can go out and buy something and put it on your field. Whether it's an organic chemical or a non-organic chemical. It's who it costs. So again, as we get more aggregation, we're going to improve porosity in the system. And so we're going to have this open space that you can see, and I just wanted to put this slide up here so you can see this illustration of this open space that's going to be between the aggregates. The advantage of this open space between the aggregates is that because of the way the aggregates fit together, I think of this oftentimes as if you had a puzzle and you basically cut off all of the little ends so that you could still see what the puzzle looked like, but the puzzle pieces wouldn't fit together exactly right. And because those pieces all have a little bit of a different structure, if you tried to draw a path through the open space, the path would bend and curve and move around through that space. The same thing is happening with water. When it comes into the surface of the soil, it's like coming in at the top of the puzzle, and it's got to bend and curve and move around and wiggle its way down through to the bottom of the puzzle. Now, the advantage of that is that between rainfalls, the sun is constantly trying to suck water out. Gravity is constantly trying to pull water down. If you have bends and curves and kinks in the path, it's going to take more energy, longer periods of time, more intense sunlight to suck the water off, or more time for gravity to pull the water down. So guess what? You can retain more water, as we're talking about with water holding capacity. You can contain more water in that area of the soil where you have good aggregate structure, because you have those bends and curves and moves around the aggregates. Similar to as if you had a drink and you had a straw in the drink and you tried to, and the straw had little bends in it, and you try and suck up the drink, and you're gonna, you go, and then you throw the straw away. That's what we need to do, is keep that water in the soil environment. Because we can't change climatic uncertainty. We can't change the weather patterns. What we need to do is create systems that are going to be able to have the resiliency to respond to those changes in weather patterns. As we build up resiliency in the system, the other thing that we're going to be able to see is ways to be able to ameliorate not just water, but also temperature. When you get this insulation cover that you're going to have on the surface of the soil, this is in an organic field, this is a rolled down cover crop. When you get that cover on there, one, it's going to help you with the bombs that rainfall brings, because guess what? The energy is going to get diffused by hitting that residue that's there first. The other thing is the intensity of the sunlight is not going to hit the surface of the soil under there very easily, because there's a blanket on it. When I was a little kid, I didn't understand this very well. We used to go to the grocery store in town, and we would buy a container of ice cream, and they used to wrap it in newspaper so that it would stay cold. And it's the middle of summer, and I would look at my grandma and I would say, why do they do that? I don't get it. Doesn't that make it hotter? You put it in a blanket. You put it in a blanket because you wanted to keep the temperature that was in there. You wanted to make sure that that was going to be protected in there. So both in the summertime, you can moderate temperatures better, and in the wintertime, you can moderate temperatures better by having this cover. So yes, you're in Saskatchewan. I understand that. But you can control how hard your soil freezes. As you build up organic matter in your soils, one of the things that happens is the freezing point of water changes. When you have soluble compounds in water, soluble organic compounds in water, it changes the freezing point. So your soils are not going to freeze as hard. And then you've got that, plus you've got a blanket on them to make sure that they're protected and the temperature is more moderated under that blanket. Wouldn't it be nice? You could grow a cover crop in November in Saskatchewan. I was in North Dakota and we were growing cover crops into December. So you need to be looking at what it is. You can't come out and say, I can't do this. One of the things that I typically ask of farmers when I give these types of talks is, one of the things I want you to come away from is thinking about your soil as your most important resource. And the other thing is to never say, I can't. Because the minute you say, I can't, and I'm going to treat you like your mother for a second, the minute you say you can't, you never will be able to. It's difficult right now. I'm not sure how I'm going to do it right now. I'm all for that. I can't quite see a path there right now, but I think I can get there. That's okay. But never tell me that it is something you can't do. I was talking yesterday about the fact that I believe that we can solve the issue with complete organic, complete no-till. I think we can do it. It's going to take work. It's not going to be an easy thing. Lots of different things are going to happen along the way, but I think that it's possible to be able to get there. So you can see that, again, as you have cover, we have cover that comes from a mulch layer. We can have cover that comes from a living plant. In this case, again, same day, this was in North Dakota, so it wasn't in Pennsylvania where temperatures can be hotter. It was in North Dakota, a little bit more relatable, right? Surface of the soil was 20 degrees cooler where we had a living plant growing. I can't change the fact that it was a 90-degree day. I apologize, this is in Fahrenheit, but I think you guys can get the idea. I can't change the fact that that was the case, but I can change the way that the soil acts and if I have living organisms in the soil or if I've killed them all. Maintaining their habitat isn't just about maintaining their structured habitat. It's helping to maintain the conditions that are going to support the life that's in the soil. So we've now been able to build a system in which we're more resilient against climatic uncertainty. Now let's see if we can build a system that's going to be more resilient to nutrient management. Because I have a phosphorus issue, right? I have a nitrogen issue. I'm going to tell you right now that you can have lots of issues. I'll give you that. You have one problem. There is one problem in agriculture, and it's a carbon problem. And it's not the carbon problem that everybody thinks it is. It's not the fact that we have too much CO2 in the atmosphere. It's the fact that we have too little carbon in our soils. You solve the carbon problem and every other issue that you have, because that's going to help you build resilience, every other issue that you have, I will guarantee you, will get solved. I'm a scientist, I don't make guarantees. It's a very scary thing. You have no idea how much I put myself out on that one. But I will guarantee you that your issues will be solved if you solve the problem. So one of the things that we have with nutrient use efficiency is the fact that our nutrient use efficiency is actually going down. So this top graph is showing the increase in production that we've had over time since the 1960s. So we've had an increase in production, done all of those types of things, worked a lot with changing varieties and crop genetics and everything, and not even going for GMO varieties, but just looking at what we're doing with breeding so that we're increasing yield. The bottom graph is what's called a nitrogen use efficiency curve. And what that means is the amount of nitrogen needed to produce a bushel, in this case, or a known quantity of grain. And so what's happening is the fact that the amount of nitrogen that is needed is actually going up. It takes more nitrogen today to raise a bushel of grain than it took in 1960. Wow. And the reason that is because we've decoupled the system from biology. We've decoupled the system from the soil that has the inherent fertility in it that the plants need. So now we have to bring all of that fertility into the system as opposed to utilizing what's already there. And although we've done a lot, again, with breeding and everything, we haven't solved this problem, we've made it worse. Because the more and more that we're selecting for higher yielding varieties, guess what? The less carbon there is available to feed the soil. It's a carbon allocation issue. I said you had a problem. We have to solve how it is that we're going to be allocating carbon in the system in order to be able to address this. So this was, again, the corn that we had in 2015. This is a close-up of the cornfield that was our conventional cornfield. And what ended up happening here is when you looked at it from far away, it looked like they were experiencing drought stress, that there wasn't enough moisture in the soil. And we'd had a lower than normal rainfall in Pennsylvania. And when I'm talking lower than normal, it all is relative. This was one of the first years I was in Pennsylvania and came from Bismarck, Mandan, North Dakota, semi-arid environment. And when they're talking about drought, they say, well, we had less than 25 inches of water, precipitation. I'm going, really? Are you serious? You have no idea what a drought is. It's all relative, I understand that. But the reality is that this isn't a water problem. This again goes back to that study that was done in the 1950s. This is a nutrient problem. So what happened in this conventional system was that they applied conventional synthetic fertilizer. We applied conventional synthetic fertilizer at the recommended rates in the spring. It was a wetter than average spring. So all of our synthetic nutrients washed away or leached out of the soil profile, because that's what happens with synthetic nutrients. When you add nutrients in a plant available form, they are highly soluble. They're going to get lost, or they're volatile, and they're going to be like ammonia or even nitrate will get converted to a gas and go back up into the atmosphere. If it's in an organic form, it's unavailable to the plant, but it is far more stable in the system. So it's going to stick around there for a longer period of time. It's not soluble in the same way. It's not volatile in the same way. So we added things in a plant available form at the recommended rates for our yield goals. Everything we did was what we were supposed to do. What we found was that as you looked up close, these plants were actually phosphate stressed. Because what happened was, although they had adequate phosphorus fertilizer in the spring, that fertilizer ran off. Or if it didn't run off, there was too much of it to be taken up by the young plant when it was there in an available form, so it became unavailable Phosphate doesn't like to stick around in the soil in an available form for very long. So it became readily unavailable. And then in the fall, it was a little bit drier. We had a little bit lower than average rainfall. Nutrients, when they are in a plant available form, to move towards the plant roots, they have to move in water films. When you have less soil moisture, the plant ends up having to give off water around its roots to try and create a diffusion gradient to get those nutrients to move towards the roots. So we ended up losing the fertility that we would have in our system normally when we were doing this. So this wasn't a water stress issue, this was a nutrient stress issue. But it all came off like it was a water stress issue. Because we weren't managing nutrients right, we also weren't managing water right. We can help the plant to control the system. And one of the ways we can do this is by working with organisms called arbuscular mycorrhizal fungi. These are organisms that are connected to the plant roots. This is their fungal hyphae that you have out there. It's going to go out there and it's going to pick up water and nutrients from the soil environment and transport them back to the plants themselves. So essentially, this is what it looks like. You've got inside, they actually penetrate inside the plant root cell wall, grow right up to the cell membrane, and form these highly branched structures called arbuscules. Now, this relationship has been around since the first plants came onto land. Since the first plants evolved on land, we've had this relationship with this fungus. Because what this fungus did was it was able to more efficiently go out there and get those nutrients and deliver them right to the plant. And what it asks for from the plant is carbon. Because it can't get carbon from the soil environment. It needs to get carbon directly from the plant. So if the plant can feed the fungus will feed the plant what the plant needs. It's a mutually beneficial relationship that you have going on here. And one of the things that happens is this fungus can go out beyond what's called a depletion zone. So right around the plant roots, the plants will absorb those nutrients. They do a pretty good job of that. But plant roots aren't really the best absorptive structures. They're a little bit too porous. And again, to move nutrients that are further away, they have to give off water so that the nutrients diffuse towards the roots, and that's not a very efficient thing to do. So what they ended up doing was they ended up helping to continue on with this relationship with this fungus that will go out there. And this fungus has the ability to obtain up to 90% of the nitrogen and phosphorus needs that the plant has. And all that it asks for is up to 30% of the carbon from the plant. Now that's kind of a scary thing, because if the fungus is giving carbon to the plant, what's going into the grain that I need to harvest? Nothing is free, as we well know. Everything costs. This is part of the decision that you're making as a farmer as to who's going to pay the cost. Because if the fungus doesn't do it, guess what? You got to go out there and buy it. Those are the choices that you have. There's no ifs, ands, or buts about it. That's just the way it goes. And you can see that inside the plant roots, this is basically roots that are stained, so that you can take a look at what's inside the plant roots. And I'm hoping to get it on our website fairly soon, but I've got a method that you can use to stain your own roots using your own household chemicals. So you can take a look at how much of this fungus you might have in the plants that you've got. But essentially, what we've got is this fungus inside the plant roots, and this fungus outside of the plant roots on the fungal hyphae, they are associated with a phosphate solubilizing bacteria. And so what happens with this is the bacteria will release phosphate when it's in an unavailable form, and it actually grows along, attached to the hyphae of the fungus, so the fungus can move it to new areas. It asks for carbon from the plant, and that allows it to be able to make the enzymes to release the phosphorus that then can get picked up by the fungus and go back into the plant. Now, this also can work with similar types of organisms that are inside the roots of plants that are rhizobium bacteria that work with legume plants to fix nitrogen. And so one of the things that we found, again, this is from the farming systems trial in 2015, I looked at the roots of our soybean plants. And it wasn't the fact that we didn't have nodules on the conventional roots, but those nodules were attached to the taproot. There wasn't very much extensive nodulation that happened outside of the taproot. So the plant itself was working with the bacteria to kind of take care of some of the own immediate needs that it had, but in the organic system, I had nodulation and much more fine root development on the organic soybeans. So now I'm not just getting nitrogen fixation that's going to help that particular plant, but I'm also going to get nitrogen that's going to be in a pool in the soil for next year's crop as well. Because I'm increasing the whole concentration of nitrogen in that soil environment. Instead of just benefiting one plant, I'm going to benefit multiple plants in that process. I'm going to skip over that slide quick. What did that happen there? Okay. So what ends up happening, and I talked about this just briefly yesterday, and I apologize because I know you've heard this story before, but you're going to hear it again because I like this story. If you have two different types of plants growing together in a soil environment, so if you have a grass-type plant and a legume-type plant growing together in the same environment at the same time, the fungus can connect the roots of those two plants together. So the roots can be all intertwined, but what happens in one root, the roots can't anneal, they can't connect each other, they can't be connected to each other from two different plants. But the fungus can provide the conduit between the two different plants. So what ends up happening is you have these nodules here where you have a bacteria that lives inside there that fixes nitrogen. And that nitrogen will go into the legume plant, and from the legume plant, it can go into the fungus, and the fungus can take some of that nitrogen and it can give it to the grass plant that is also associated with that particular fungus. Now, to fix nitrogen, it's a very energy-intensive process. What you need for every molecule of nitrogen gas, N2 gas, you need 32 molecules of what's called ATP, or adenosine triphosphate. It's supposed to be ATP. Adenosine triphosphate. What happens is to get energy, you have one of those phosphate groups that gets ripped off, and it releases electrons, and they go into the electron transport chain, and that basically fires the battery, or it fires the cell, so that the cell has energy to do this process. When you take that phosphate group and rip it off, you can't put that same phosphate group back on because it lost energy in that process. So you have to have a new phosphate group come in. So essentially, to do all of this, you're going to need 128 phosphorus atoms and 320 carbon atoms to fix one molecule of nitrogen gas. It's an energy-intensive process. That's why nitrogen fertilizer costs as much and produces as much greenhouse gases as it does. This just happens on a cellular level. You can't escape it. It takes energy. There's no way around it. This is the way that it works. Now, to get that much phosphorus, the bacteria in the roots of the legume ask the legume to give it phosphorus. The legume can't really get phosphorus very well because its roots aren't very good at absorbing phosphorus, and phosphorus isn't very available. So it goes in and asks the fungus to help it to get the phosphorus that it needs. Now, the fungus has the advantage because the fungus is associated with the phosphate solubilizing bacteria that are on the fungal hyphae, and that can help to release the phosphate. But now that you got the phosphate in there, the fungus can only absorb so much phosphate, and the demands on the part of the legume, as well as the demands on the part of the grass, as well as the demands on the part of the bacteria in the legume, are very, very high for that phosphorus. So it can be very difficult for the fungus to be able to absorb enough phosphorus in order to be able to do this. So what the fungus will do is the fungus then goes and it tells the grass, I don't really know how to do this, and I've had this relationship with you for a long time, and I heard that you know the secret code to tell me how to absorb more phosphate. So the grass gives the fungus the secret code to absorb more phosphate so that it can absorb phosphate, it gets degrade it gets the phosphorus from the bacteria that colonizes its hyphae so that you solubilize that phosphate, the phosphate goes into the fungus will then give some of that phosphate to the corn says, I also want nitrogen. Then the fungus says, all right, then I'll go over here to the legume and get nitrogen, but the legume says, well, if I'm going to give you more nitrogen, you're going to have to give me more phosphorus, so it can get that phosphorus, and that's going to give that to the legume, but the legume's going to give that to the bacteria. We've got 5 organisms, 2 bacteria, 2 plants, and 1 fungus. They do this all the time. This is what they do. This is their job. This is how they get fed. This is one particular relationship grouping that we know about. It's not beyond conception to assume that that doesn't happen with a whole bunch of other nutrients that are out there. This is just one that we know about because we did some labeling studies to figure it out in the laboratory. So the soil has this inherent ability to build the resilience to resist the stress that's going to happen with nutrient management issues if we allow it to have that occur in that system. So now we're also going to build resilience to help with pest management. And if we're looking at weed pest management in the system, what we want to do is we want to reduce the number of weed seeds that you can have in the system. You want to reduce weed seed germination. We found that there are certain types of compost mixes that may help you reduce weed seed germination, so we're doing some research with that. We want to increase the amount of competition that you're going to have in the system. We want to utilize some things like rolling down a cover crop or mowing a cover crop down. We want to utilize strategic tillage. So we want to do this in a reduced tills type of system, or we could utilize grazing to help us manage weeds. And so we're doing research on looking at all of these different aspects at the Rodale Institute. And one of the things that I do want to talk to you a little bit about is the whole idea of shifting the paradigm in your mind that the default tool for managing weeds is tillage. Because just in the same way that you get upset at a conventional farmer whose default tool for managing weeds is a chemical herbicide, you're not shifting away from the paradigm that your default tool for managing weeds is tillage. Is still destructive to the system. So let's see if we can move away from our default tillage tool. We've seen in the US an increase in the amount of erosion. And as we've gone to more sustainable management, as we've gone to conservation tillage and those types of things, we've reduced the amount of soil erosion that we had. But the reality is this is a 2007 number. I saw a 2014 number that was essentially the same number. And we're happy with this. This is a sustainable system. This is what we're promoting. Sustainability, right? We can sustain losing 2 billion metric tons of topsoil on an annual basis. We're pretty rich in this continent. We can be here for, a few hundred years at least. I don't know where we're going to go after that, but hey, we can do it. Or we can try and do other types of things. We can look for changing how it is that we're going to be managing the system. So at the Rodale Institute, we developed a tool that's going to allow us to be able to roll down a cover crop. And basically what this tool does is that it doesn't mow or slice or cut the stem. What it does is it breaks the stem in a number of different places. And the advantage of this is that many plants, when there's carbon resources in the roots that can then be released from the roots to the organisms in the soil to get minerals so that the plant can produce the proteins and part of the plant's physical structure that's needed, the plant has that ability to do that. So if you cut it, it doesn't necessarily kill it. But if you break the stem in a number of different places and you break the stem at the right time, and when you break the stem at the right time, it means that most of the carbon resources are up above ground in the flowering parts of the plant. So there's less carbon in the roots. so that when you break the stem, you put a whole bunch of wounds, and on the other side of the wounds, on one side of the wounds is the carbon resources that are needed to make the proteins to seal off those wounds, and on the other side of the wound is all of the mineral sources that are needed to make the proteins to seal off those wounds. Guess what? It's pretty tough for you to be able to get those two things together to make the proteins to seal off all those wounds. So that's what we're trying to do, is identify different types of cover crops that we could use, that we could effectively roll and be able to have that killing of the plant, so that our cover crop doesn't become a weed issue. And since we first created this tool, there's been a lot of different types of this tool that have been developed over time. We've got a lot of different shapes and sizes and uses for this particular type of tool. We've got things that you can use if you've got a very large fields. We actually did some work with Howard Buffett, who has two 60-foot rollers, and they're with his no-till drill. And so they've got the arms to fold up. It's attached to his planter. He had it custom made so that it folds up in the same way that the arms on his drill fold up, so that he can drive it down the road. So you can get it very large. You can get it to be used by a bunch of different types of equipment. We've got them, you can have horses that are going to be able to pull them. So they're used by the Amish community. We've got ones for raised bed type systems. We've got ones we can use in vegetable systems. And we had one young lady who actually decided to put a cover crop in her high tunnel, and talked to her dad about it, and they took a piece of angle iron and attached it to a 2x4, and then she put ropes on each end, and she pushes it along with her foot and just kind of scoops it up to be moving it along, and terminates the cover crop that way, rather than having to go in there and do any type of hand weeding or anything like that. It reduces the amount of hand weeding that she's got to do in the system. The other tool that we've got that we use on occasion is a high residue cultivator. This cultivator, this piece of equipment, has very wide sweeps, and what we can do is that we use it with our roll down cover crop. The sweeps will go in. They basically have a little bit of a sharp blade here that slices that residue so that the sweeps can get tucked underneath it, and it will basically tug and pull the weeds out right at the soil surface. So if we have weeds that are coming up in our residue that we rolled down, if our layer we didn't get enough biomass, we can take this high residue cultivator out there. Do very little disturbance to the soil, because it's right at the soil surface that it does this. We do get a little bit of disturbance to the soil underneath, so you'll get a little bit of disturbance, but it only goes down about less than 1/2 inch. So minimizing the amount of disturbance that we're doing to that soil environment. We can utilize competition in the system and be able to have companion crops. Companion crops and cover crops are you picking the weed, essentially. Nature picks it or you pick it. It's your choice. Again, it's like nutrients. It's your choice. Somebody's got to pay for it. Somebody's got to cover the soil. You can choose to cover the soil with something that's going to benefit you and be easier for you to manage, or nature can choose something that's going to make you mad. Because nature does that, right? Nature's out to get you, right? Every organism is living for its own sake. This isn't a thing that somebody's out to get you. It's a thing that's making the system function better. So we can use different types of cover crops that are going to help us with getting more nutrition, having different types of mixtures in there, utilizing nurse crops to help us get some of our rye or oats start growing and be able to cover the soil, making sure that you have something growing as much as possible in the system, making sure that what it is that you're choosing isn't going to interfere with any type of your harvest operations. It's an important thing to be thinking about when it is that you're picking the types of cover crops you're going to use. We're also trying to do some things with breeding weed resistance in the system. We're trying to create a system in which we're actually looking at selecting for plants that are going to be able to tolerate having some weeds in there, that they're going to outgrow and outcompete the weeds that we would have in that system. Again, nature will pick your cover crop for you, or you can pick it. We don't want to necessarily get rid of some tools that could actually help us in the system. Skip over a couple of these slides. So we want to be able to make sure that we're reducing the amount of weed expression that we're going to be getting in that system. And we can use a number of different tools. You can use insects to be able to manage weeds in your system. They're going to eat weed seeds. Can utilize small rodents to help to manage weeds in your system. All of these things are viable options for us to look at. We also want to help to manage insect pests that you have. So we utilize things like predatory insects in the system. We're going to be utilizing trap crops and insectary strips to enhance the amount of predators, insect predators, that we have to manage that. We're also utilizing a compost-based seed coating. We're doing some research on this, where rather than in the conventional world they have a fungicide seed coating, we're going to use a compost-based biological seed coating. We're going to put on the seeds and reduce the amount of soil-borne pathogens that we would have in the seedlings in our greenhouse that we would then use for transplants in our vegetable cropping system. So prevent the amount of root crop diseases before we get to that. So we're going to be looking at really rebuilding and regenerating the soil in that system. And that's what it is that we want to focus on, is getting the soil to be able to function for us and utilizing all of these biological practices to help build resilience. Again, I'm a scientist. It's a difficult thing for me to say, but I can guarantee you that if you solve the problem, all of your issues will go away. And that's what it is that we need to do, is regenerate the soil, regenerate that carbon to solve that problem. So thank you.